Hi guys, and welcome to Chapter 10 Part 1 video notes. So we're going to start talking about the molecular biology of the gene. So we're going to be talking about DNA. So in class we started talking about Griffith's experiment on the mice, and some of them sadly had some bad outcomes. So first we discussed virulent pathogenic bacteria and non-pathogenic bacteria. So that pathogenic bacteria is disease-causing and the non-pathogenic is not disease-causing. So the first mouse that he did his experiment on was injected with non-pathogenic bacteria, and the mouse got to live. The second mouse here, he wasn't so lucky. He got injected with a pathogenic bacteria, and this killed him. Then what Griffith did was he took that pathogenic bacteria, heated it up really hot so that it was destroyed. The DNA was still there, but it was destroyed. And since the cell was destroyed, it could not affect that mouse, so when it was injected, he lived. And then right here it shows that mouse being injected with a mixture between the non-pathogenic bacteria, the non-disease-causing bacteria, with the heat-killed pathogenic bad bacteria. And what happened is the mouse died. So let's take a look at why that was. So 10.1, Griffith found a discovery about the transforming factor that transferred into a bacterial cell. So what he did was, we talked about how he took that disease-causing bacteria and heated it up to kill it. Okay? So what that happened is some harmless cells that were inserted into that mouse with that heat-killed bacteria were converted into disease-causing bacteria, pathogenic bacteria. And what we call this process is transformation. That's a very, very important word, transformation. So that harmless, non-pathogenic bacteria slurped up that DNA from that pathogenic bacteria. So it slurped it up and became possessed and was a bad bacteria. So one more time, look at this picture. The mouse was injected with a non-pathogenic bacteria and lived. A pathogenic bacteria and died. Heat killed pathogenic bacteria and lived. And then heat killed pathogenic bacteria and non-pathogenic bacteria. And that non-pathogenic bacteria slurped up that bad DNA and killed the mouse. So then we talked about Hershey and Chase, and what they did was they did an experiment on bacteriophages. So, review, bacteriophages are viruses that infect bacteria cells. We call them phages for short because nobody wants to say bacteriophages all the time. So what they did was they labeled these phages with radioactive sulfur to detect proteins, because remember that sulfur binds to the protein, or radioactive phosphorus to detect DNA, because remember DNA has phosphate in it. So that phosphorus will bind to that DNA, and it's like a label. It will label whether protein or DNA shows up. So then the bacteria were infected with these viruses that were labeled with this radioactive sulfur or radioactive phosphorus. And they did this to determine what substance was injected inside of the bacteria. Because remember, taking a look at a picture here, that virus sits on top of that bacteria cell and injects genetic information into that bacteria cell. We know they inject DNA, but Hershey and Chase were still learning this. So they did this experiment to determine whether this virus shoves in protein or shoves in DNA, because that was the big question. So what the experiment showed is that the sulfur-labeled protein stayed outside of the bacteria cell, while the phosphorus-labeled DNA was seen inside that bacteria cell. So this is a really, really important discovery. So let's take a look at the picture. So batch 1, radioactive protein is put in with those viruses. And you see those viruses, the outside is yellow. That outside protein coat is yellow. They mixed it all up into a blender so that way all the viruses were forced off the bacteria and they could just look at the bacteria. So then in the end, at the very bottom here, we have what's called a pellet. These are just the bacteria that sink to the bottom, and then all the viruses float to the top. So you notice that the viruses floating at the top, that's all yellow. So then when they looked at the cell, they didn't see any radioactive sulfur in the cell whatsoever. So they could then conclude, well, it's not protein that's being inserted into this bacteria. So then the second batch, they took radioactive DNA. So they took that phosphorus labeler and put it inside with those viruses. They mixed the viruses up and the bacteria so that the viruses would detach. And then when they took a look at the bacteria, what do you know? There's radioactive phosphorus inside that bacteria. See that green in there? So they knew that DNA is what was inserted from the virus. And then they were very happy because they knew that the genetic material was DNA. So let's talk a little bit about DNA and RNA. So they're polymers of nucleotides. So let's review real quick. A polymer is like a big unit, and then a monomer is the smaller subunit within it. So if I had a long chain of pearl necklaces, that would be my polymer, and then each pearl inside the necklace would be a monomer. So the monomer unit of DNA and RNA is the nucleotide. So that nucleotide contains a nitrogenous base, a 5-carbon sugar, and a phosphate group, which should be reviewed from earlier on in the year. So DNA and RNA are polymers, so that they're that long chain of pearls, and they're called polynucleotides. So they have a sugar phosphate backbone, and it's formed by covalent bonding between a phosphate of one nucleotide and the sugar of the next. And it'll make a lot more sense when we look at a picture. Then the nitrogenous base extends from the sugar phosphate backbone. So they're kind of pushed into the center. So let's take a look at a picture. Okay, so here's DNA. It's stretched out to lay flat for the picture. Okay, so here's our DNA nucleotides. Okay, our sugar phosphate backbone. So each one of these is representative of a sugar. So we have our sugar backbone. These little yellows represent phosphate groups. And then we have our nitrogenous base. So we have this A, C, T, and G are all nitrogenous bases. So then let's take a look at one nucleotide blown up. So here's our phosphate group, our sugar, and our base. So there's that blown up. If it helps you, I would draw it just to kind of get as a reminder to you what that nucleotide looks like. A DNA nucleotide, the structure is very, very important. So the pyrimidines, so we're looking at this nucleotide base, okay? This DNA nucleotide. We're blowing it up here, and what we're looking at is the 4 different possibilities. The first possibility are called pyrimidines. Pyrimidines can either be thymine, T, or cytosine, C. Then the purines can either be adenine, A, or guanine, G. So, before we go into that, that's just our background information. Watson and Crick didn't know what the structure of DNA looked like. Everybody wanted to know, okay, now that DNA is the genetic material, we need to find out what it looks like. What's the structure? So Watson and Crick, they deduced the secondary structure of DNA using X-ray crystallography, and they actually stole that information from Rosalind Franklin. It's kind of a sad story. So Watson and Crick are out to find the way that DNA is structured. So what they found was DNA is composed of two polynucleotide chains joined together by hydrogen bonds. So remember, hydrogen bonds are weak bonds, and it's twisted into a helical shape. That's what they figured out, is that helical shape. So I'm sure you guys know that DNA is a double helix. So the sugar phosphate backbone is on the outside. Remember, we have our sugar backbone in DNA. The bases are perpendicular to the backbone inside the interior. So we have our DNA that wraps around, and those bases stick inside towards each other. And these specific base pairs, remember how we talked about the pyrimidines and the purines, those specific pairs give that a double helix shape. So something very important to write down and to remember is A pairs with T and G pairs with C inside DNA. And how I always remember that is G kind of looks like a C a little bit, so G and C go together. So here's a picture of Rosalind Franklin, who found this picture using crystallography, which none of us probably would know, like, what in the world that looks like. But she knew. She knew, looking at that picture, that was a double helix. And Watson and Crick stole that from her. So here we go. So here is our hydrogen bonding between our bases, A with T, G with C. Here's our sugar and our phosphate, the three parts of that DNA. So when it's all together, you see how DNA forms that double helix. And it's those bases that make it twist to form that double helix. And here it is again. There's a double helix. You see that A pairs with T and C pairs with G. So that is all for part 1 of Video Notes. I hope you enjoyed.